Okay, so my name is Dean Bushey. I'm going to try to walk you through an introduction to Task Force 11, which is the title of the paper that we're working on is Characteristics and Safety Assessment of Intractable Proteins in GM Crops. So the mission that we were assigned or we took on in this task force is something I think most people in the task force felt very attached to, because we knew it was a real problem. It wasn't a theoretical problem, and a number of people in the task force have already run into issues with this. So the mission is really to address the question of how to perform a science-based safety assessment of intractable proteins that are difficult or impossible to purify, isolate, or characterize from plants or recombinant systems used to express these proteins. So it's a fairly general phenomena, and it runs across a lot of proteins. And the first thing we had to do when we were going to attack this problem and come up with solutions to it is how are we going to organize ourselves? And this is before I get to that, let me just mention a team here. I won't go through everybody, but it has academics, academic, I should correct myself, Jay Thielen, which has been mentioned many times before. Was on our committee, very, very helpful. We had Mary from Canada and Gloria from Mexico participated from the agencies and appreciated their help, and a number of people from the different companies. And I should mention here, Kate is not on this thing, but Kate did herd everybody together, and sometimes it was like herding cats at times, so we appreciate her help from that. So how did we organize the task force? How did we get started? We started off by thinking about, okay, if we have intractable proteins, what kind of issues are we talking about here? What describes an intractable protein? Because, well, to be honest, we spent a lot of time coming up with the word intractable protein. So once we came up with a word that nobody knows what it really means, we had to define it first. So the issues these are the four issues that These are the four issues that we really concentrated on. For the issues associated with intractable proteins, it's suitable expression system. So could you find an expression system to express this protein if you needed to make large quantities of it? Some of the proteins that we talk about intractable are there because the low level of expression of protein. And Dr. Parrott just mentioned transcription factors, that's one of them. Some of those are incredibly low levels. So how do you do studies on proteins that are expressed at low level? Another one is functionality of the isolated protein. You may have the protein and maybe they're in quantities, but when you try to isolate and purify it, is it really still functional? So when you take it out of its microenvironment, you lose all the activity that you had, the functionality of it. And the last one is the expression in a different system. Okay, these proteins are expressed in plant. To make large quantities to do the typical safety assessment, you have to put it in some other system. And the question is the equivalent? Are you making an equivalent protein when you're working in another system or not? So we came up with those issues that we wanted to deal with, and we agreed upon that. And then we decided okay, if these are the issues, what are the types of proteins? How can we sort of get this idea across in a paper? So what are the types of protein classes that we can use to demonstrate these issues? And to do that, we again came up with 5 different classes of proteins: membrane proteins, signaling proteins, transcription factors, N-Glycosylated proteins, and resistant proteins. These are, it's not an exclusive list, but it's a list that we felt was a good list of proteins that we can use to demonstrate the issues of intractable proteins. So when you do that, make that list, and you start looking at which issues are demonstrated by which protein classes, you come up with this type of arrangement here, where some classes have all the issues associated with them, all 4 of them. Some of them only have 2 issues. What we try to do is, let me just show you, this slide is a little easier way of looking at the same thing that we just showed you, that the membrane proteins have all 4 issues associated with them, signaling proteins, 2 of them, etc. There was some discussion, I must admit, at the beginning of the paper that this should really be a task force on membrane proteins, not intractable proteins, because membrane proteins are probably the most important class of proteins here and the ones that people are most worried about, I guess, on how to do the safety assessment of. We decided to expand it beyond membrane proteins to include these other types of proteins, just because we wanted to expand it and not limit it to one class of proteins as if that was the only issue that you needed to deal with. Because all these proteins are going to have this issues, and all of them are intractable in one way or the other. And we tried to use in the paper, we divided up into two sections. The first section of the paper is really to explain intractable proteins, what we mean. Definition of the intractable protein. Each class is used, the protein classes, we used each those classes to demonstrate an issue of intractability, of what we mean. And we tried to give some specific real-life examples in each one of these classes of what an intractable protein in that class might look like or why it's intractable, so that we aren't talking about theoretical issues here. We're talking about real-world problems, and we wanted to make sure that that came across. So let me, I've just picked out 3 examples from the paper that's in the draft that we have right now, and I'll go through those to show you what we were trying to do. And remember, this is the first half of the paper where we're still defining intractable proteins and what the issues are. So the first one would be a membrane protein, and there we use this diacylglycosyl acyltransferase, DGAT, as an example. And it's pretty obvious thing when you try to, this is a membrane-incorporated protein. As you try to purify this protein, obviously you lose activity. So the purer the protein, the less activity you get. By the time if you ever could get a pure protein, you would have a non-functional protein because it's not in the membrane, and the membrane is part of the activity required. So that's the big issue with all membrane proteins, and this is a good example of that. The other issue is if you did try to make this protein in another system, and it has been made in SF9 cells, insect cell lines, the levels that you get are 1 to 2 milligrams per liter, which is a little bit difficult if you're trying to generate grams of protein to do your typical toxicity studies that have done on a lot of the proteins. So again, we go back to low levels of expression is a problem with some of these proteins. And this is another history of safe use. I mean, we use that a lot. This is a way to deal with some of these things because this protein itself has been out there and has been consumed for many, many years. So this is on membrane proteins. We use this as an example of how to do functionality, how to demonstrate functionality of isolated proteins. Another class is the N-Glycosylated proteins. And we sort of use this class to this issue of equivalency, and this was mentioned before. If you're going to express this in a different system, do you have an equivalent protein or not? And the problem here is this protein we're talking about here is a single chain, but it gets proteolytically processed into 2 subunits, and each of those subunits are glycosylated. And if you take this gene from a common bean and you express it, let's say you're using P or some other system as an expression system, you express this in a P, the N-glycosylation pattern is not the same in P as it is in bean. So do you really have the same protein and if you express it in a different system? And it's interesting that the authors comment in the paper that looks at this concept that even within bean, there is a difference between bean being expressed in P, but that difference, there's more of a difference between bean species as far as the glycosylation, the sugar pattern, and the amount of the proteolytic processing that actually happens. So even within beans, different species of beans, the question is what protein are you really going to be looking at here? If you want to really do a safety assessment on a single protein. There is no single protein in N-glycosylated proteins. It's a range of proteins. It's a population, basically. Another example that we have in a paper is this of an R gene. And this is some interesting numbers here. This gene confers resistance in to late blight disease. If you look at the limits of detections of this, for this protein, it's about 100 ppb. And if you do a calculation, and it's been assumed that this protein exists maybe in 1 copy per cell, could be as low as that. And if you do the calculation, if it is in 1 cell, 1 copy per cell, and you do the calculations on how many cells in a leaf and et cetera, you come out with at the concentration of this is probably about 0.018 ppt. So you can see where even if the calculations are off by 100 or 1,000, the level of detection here is above the level that is actually in the cell. So it's, I mean, how do you do an equivalence study of that? And even to get the point across, it's an interesting calculation here. If these calculations are correct, and you do the normal dietary exposure, the calculation comes out, you're exposed to about 3.6 nanograms of this protein per year. So we do introduce into the paper, the question is this really where we want to be spending our time and energy to be doing a safety assessment of a protein that's expressed at this level? And I think that's something that needs to be considered when looking at some of these low level expressed proteins. So that's the first half of the paper, sort of the different protein classes and the intractable issues, and hopefully we make a real case that it's a real issue and it's something that has to be dealt with. So now the second half, the real meat of the paper becomes, okay, now that you've defined it and you believe that there is an issue out there, how do you deal with it? And for this, we've really went back to task force mostly like 6 and 10. So we try to build upon what because I think they've established a good basis for us to start from. And we went back and looked at Tier 1 tests and Tier 2 tests that Task Force 6 introduces, and looked at the different studies. And we go through in our Task Force 11, we go through these examples or methods and tests that could be used for an intractable protein. None of them are going to be, some of them may not be perfect. You may not be able to do the perfect study with an intractable protein, but some of them you don't even need protein. Obviously, the bioinformatics, history, safe use, some of those are just as relevant to an intractable protein as a tractable protein. Other ones like the omics has been mentioned before. We spent a lot of time talking about what's the role of omics in a study like this. We think if it's directed and targeted, we think that omics does have a role to play, but it shouldn't be just an open-ended shotgun omics approach, because that brings up more questions than it does answers. But if you know the mode of action, and you know what you're looking for, then omics does have a role to play, we think, as long as you realize what the restrictions are. If you have any issues at Tier 1, or if you think you know to go to Tier 2, again, there's studies in Tier 2 that we think you could do, modified from the normal studies that you do with a regular protein, where instead of doing the protein, you might do microsome preparations, you might do a semi-enriched fractions. Again, it's not the ideal situation, but it's probably pretty close to the numbers you're going to get out are probably pretty real and probably pretty reliable to use. So we do go through a number of studies that we mentioned and talk about suggestions for what might work for an intractable protein at the Tier 2 level, if you need to go that way. So the key messages that we want to leave at the end of the paper, the first one is intractable proteins represent many very valuable traits for future development. This is something that there's a lot of these in the pipeline today we know about, that people are working on them, and it's only going to get more and more as the future goes. I mean, herbicide tolerance, insect resistance are good traits, but these are the next generation, and there's a lot of possibilities out there that's being developed now. The other important thing is a safety assessment can be done on an intractable protein. We don't want to leave with the thought that, okay, it's an intractable protein, we can't get the purified protein in our hands to make a couple grams of it, so forget it, you can't do a safety assessment. You can. We think with the right tools, adapting the right studies into a new format, we think you can do a reasonably good assessment of that. The other thing is many studies can be done without the isolation of pure protein. We know that you don't need a pure protein for all the things that you're going to do. And I think the key here is that no single study is likely to perfect or sufficient to say a protein is safe. So where you might do a toxicity study with a protein that you've isolated and purified. I think in the intractable proteins, it's going to be hard to come up with one study that says, yeah, this is a definitive study that says this protein is safe. But you can generate multiple studies. I mean, if you have this study, and you build on, and you build a weight of evidence, I think you can build the studies together to say that this protein is going to be safe. And then again, that goes back to the weight of evidence on multiple studies will support a safety assessment. So I think that's the message we want to leave people with. We hope in the paper that we give them enough tools. There's no way to take somebody and tell you exactly how to do it because each case is going to be different, but we tried to lay out in the second part of the paper the tools that could be used to make a good case for a safety assessment of a protein. I would say, I think that's, yeah. Where we are right now is we've gone through the rough draft, the part of the writing a paper like this that really is painful, I'd say, and the wordsmithing and all that. So we've done the internal review with the committee. We've rewritten a paper. We've done the internal review with the committees, the organizations that the committees represent. We've rewritten that, and now we have it to the point where we're ready to send it out to external reviewers to get their input back from that. So we're anxious to see how that goes. If there's anybody here that wants to volunteer to be an external reviewer, we'd certainly accept. Kate sitting back there would love to have your name and you volunteer to do that. And Laura and Gerson are in the audience. We'd help write this paper, so if there's any questions, they would be glad to answer your questions for you.